All right, what's up, you guys? So if you've been following along with lecture, you've noticed that directionality in DNA is super important. So we're going to take this video to make sure that we're on the same page about everything. Right, so we've heard a lot of 5', a lot of 3'. Let's make sure we're on the same page of what that means. Right here, we have a nucleotide. And right now, check in and pause and see if you can point out what is the 3' end and what is the 5' end. All right, welcome back. So the nucleotide, I always remember it like the 5' end is the phosphate group, right? They kind of make the same sound. So this guy right here is the 5' end, right? It just means the end of a nucleotide where there's a phosphate, and the 3' end is on the alcohol, right? Okay, cool. So now we can move forward now that we know that. Just some important things to know about DNA that I want to make sure we're on the same page on. Right, it's double-stranded. You guys already knew that though. Probably even before this class, right? It's double-stranded. It's complementary. We'll talk about what that means, and it's anti-parallel. We will also talk about what this means. Right, so let's say here's our double-stranded DNA. So one strand, another strand. Just a quick quizzing you guys moment. Do you guys know what kind of bonds link the two strands together, right? Go ahead and pause and check in if you know what kind of bonds these are. Okay, welcome back. These are hydrogen bonds in a base pairing, right? So in the basis pair, we're talking about hydrogen bonds. Not related to the question, but just a quick moment of making sure that you're quizzing yourself as you're going through the material. Cool, so it's double-stranded, check. It's complementary, and what does that mean, right? That means that for every A, it will be hydrogen bound to a T on the other strand. For every C on one strand, it'll be hydrogen bound to a G on the other strand, right? So we can draw that in our little diagram here where if there's an adenine here, there'll be a thymine here, right? Every C pairs with a G, right? So that's what complementary means. Cool. And then it's anti-parallel, right? So it's parallel because they're aligned like two parallel lines. The anti-parallel part, that's when we start thinking about directionality, right? So if this is the 5' end of one strand, that means that the opposite end of the strand has to be 3'. So can you point out right now, this end of the opposite strand, is that the 5' end of the opposite strand or the 3' end of the opposite strand? Right, that would be the 3' end of the opposite strand. Cool, and this will be the 5'. So this is what we mean by anti-parallel. So it's parallel because it's parallel lines, but the anti part is because the 5' end of one strand will be the 3' end of the opposite strand. And this will follow us basically for the rest of the class. So it's important to make sure we're on the same page. Cool, so I will leave this here because it'll help us for this question. So now that we know all that, we can finally tackle this guy. Let's use a different color. Cool. Right, so we're going to talk about the template strand. So the template strand, right, whatever DNA polymerase is using as a template strand, that's the strand it's actually copying off of to make the new one, right? So it's copying off of the template strand, and it's reading the template strand, right? It's reading it and saying, what should I put into my new strand? And so the mnemonic goes, you can only read for 2 hours, right? You can only read from 3 to 5. So that's the direction it moves on the template strand that it's reading, right? You read the old strand. Right, so let's go ahead and say that in this specific case, let's say that the top strand is the template strand. If this was our enzyme doing some copying, it would be moving from 3 to 5 on the template strand. What does this mean about the new strand? Well, the new strand, right, if this was really DNA replication, this strand wouldn't exist yet, would be being made. Right, the new strand would be being made in this direction, right? Let's say this new green line is the new strand. If so, what end of the new strand is this? Is it the 5 or is it the 3? Everything is still anti-parallel, so this would be the 5, and this would be the 3. So this means that DNA polymerase would be writing, right, or new strand creating or writing You'll be writing the new strand in the opposite direction, the anti-parallel direction, right? So be writing from 5 to 3, right? So that's one way to think about it. Definitely, you know, make sure that that information is in your brain for the rest of this class. Another way to think about it, I like to give you guys more than one way to think about it, is to use some information you already know. So let's look at this nucleotide. I'm going to get rid of this text. Right. Making a new DNA strand is all about linking these nucleotides together. So you have a bunch of them together, right, making a really long strand. And one way that's helpful to think about it is, remember when we were learning about dehydration reactions, I said you guys could add a monomer to the alcohol. Right, I find that useful when thinking about this, right? So when you look at this nucleotide, where could you add another nucleotide? Right, you could add it to the alcohol on the 3' end, right? So let's go ahead and just elaborate on that, right? So we could add a nucleotide here. Then we could add another nucleotide, keeping on adding them to the alcohol, which is the 3'. So it's growing, the new strand is growing in this direction from the 5, from the phosphate group, 5' the phosphate, growing all the way to the 3' end. Right, so 2 ways to kind of think about the same thing. And if this video helped you out, I recommend trying this question now on your own before you watch the video.